The whole which came first, the chicken or the egg thing.
Exactly. But what if I told you that modern science has actually finally broken the loop?
It's wild because we've treated this like a children's riddle for so long, or, just a philosophical thought experiment about infinity, that we completely forgot it might actually have a tangible scientific answer.
Yeah, and today's deep dive is going to give you that definitive answer.
Hmm.
We are pulling from a really massive stack of sources today, like everything from ancient zoology texts and marine biology press releases, all the way to some incredibly dense, highly technical structural biology papers.
And the core thesis we found across all these really diverse sources is absolutely fascinating. Because, well, the answer to the riddle completely changes depending on which scientific lens you actually look through.
Right, so our mission today is going to be a bit of a journey for you. We are going to travel back 1 billion years in evolutionary time. Then we're going to zoom way, way in on the microscopic crystal structures of a modern eggshell.
And then finally, we'll discover how the answer to this ancient riddle might actually solve a massive crisis in modern human medicine.
Which is just crazy. It kind of reminds me of asking, what came first, the blueprint or the house?
Oh, that's a good way to look at it.
Right, because you naturally assume they just go hand in hand. But what if you discovered the blueprint was drafted 1 billion years before the house was ever built?
Yeah, that is a remarkably accurate way to frame what we are about to explore. Because to figure out who wins here, we really must first decouple the two halves of the riddle. We have to strip away the chicken part of the equation entirely and just look strictly at the evolutionary history of the egg.
Okay, so let's trace the timelines then. If we look at domestic chickens, which descend from the red jungle fowl in South and Southeast Asia, archaeological evidence and DNA show they really haven't been around that long, right?
No, not really. They only appeared roughly 10,000 years ago.
10,000 years, okay.
Yeah, and in geological time, 10,000 years is literally just a blink. Now, compare that to the egg. Hard-shelled amnio eggs date back about 350 million years.
Okay, 350 million. And real quick, an amniote is...
It's essentially any land-dwelling vertebrate that lays an egg with a special internal sac, the amnion. It's what keeps the embryo from drying out when it's on dry land.
Got it, okay. So way before chickens.
Oh, way before. And if we look specifically at dinosaur and early bird eggs, like those of the ancient Archaeopteryx, we're looking at a timeline of like 150 to 195 million years ago.
Wait, let me just push back here for a second, though, because when we say egg, you and I are usually picturing that, hard white shell you crack on the edge of a frying pan.
Right, the classic breakfast egg.
Yeah. But what about before hard shells even evolved? Or, what about before animals even had the physical anatomy, like an oviduct or an egg tube to actually lay an egg? Are we really still calling those eggs?
Well, that is the pivotal question right there. And answering it takes us much, much further back, because the fossil record actually shows ediacaran eggs dating back 600 million years.
Wait, 600 million?
Yeah, these are tiny sphere-like specimens found in ancient seafloors, existing well before complex animals as we understand them had even evolved.
Wow.
But the true billion-year twist in our sources comes from a 2017 discovery in marine sediments around Hawaii. It involves this microscopic organism called Chromosphaera perkinsi.
Wait, a microbe? How does a single-celled organism from Hawaii have anything to do with the chicken egg?
It's crazy, but it reveals the origins of the actual biological machinery needed to make an egg. This microbe separated from the animal evolutionary line over 1 billion years ago.
Okay.
And what scientists from the University of Geneva observed is that once these single cells reach their maximum size, they don't just split into two identical cells like a typical bacterium would.
Oh, they don't. What do?
They divide internally, without actually growing any larger.
So wait, instead of splitting into two separate microbes, it just stays one size but chops its insides up into smaller and smaller rooms?
Precisely. Yeah, it forms a multicellular colony. And the specific three-dimensional structure they form as they chop up that internal space perfectly mirrors the early stages of animal embryonic development today.
No way! So it's like watching a single-celled organism just suddenly start building an animal embryo.
It is. And they sustain this state for about a third of their life cycle, comprising at least 2 distinct cell types.
That is wild.
Right. It basically implies that the complex genetic programs governing embryonic development, the fundamental genetic tools to, quote-unquote, make an egg, they were already present and functioning 1 billion years ago in the ocean.
Long before nature invented chickens or egg tubes or even complex animals at all.
Exactly.
So if we look at this strictly from, a Darwinian evolutionary standpoint, Somewhere in the ancient jungles of Southeast Asia, a bird that was very, very close to being a proto-chicken laid an egg. And inside that egg, a genetic mutation occurred. The exact moment that the sperm and the egg met, the DNA of the very first true chicken was defined.
Yes. The boundary line of the new species was crossed at the moment of fertilization, right inside the egg. Therefore, evolutionarily speaking, the egg wins by an absolute landslide.
Right.
The concept and the biological mechanisms of the egg existed 1 billion years before the chicken. And the specific genetic combination that makes a chicken happened inside an egg that was laid by a non-chicken.
Okay, so if we zoom out to the widest possible evolutionary lens, the egg is the undisputed champion. It's not even a contest.
Not even close.
But this is where the story takes a massive U-turn, because if we put away that evolutionary telescope and pull out the structural biology microscope, the narrative just completely flips.
It does. And this is where we have to narrow our scope from an egg in general to the chicken egg specifically.
Right, the specific hard-shelled thing.
Yeah. So in 2010, researchers from the universities of Sheffield and Warwick tackled this by looking at the actual molecular mechanism of eggshell formation. They used this massive supercomputer called Hector to track exactly how a chicken eggshell is formed.
Firing up a massive supercomputer just to watch a shell form seems, I don't know, incredibly intense. Why did they need that much processing power?
Well, simulating molecular dynamics at the atomic level across millions of interactions takes immense computational power.
Okay. That makes sense.
Yeah. They were looking at the transition of calcium carbonate inside the bird's body. Initially, it's amorphous-like, it's a liquid-like mixture, lacking any defined crystalline structure.
Just a mush of calcium.
Pretty much. But the supercomputer allowed them to identify a crucial protein acting upon it, called ovoclidin-17, or just OC17 for short.
Wait, how does a soft protein actually force a liquid mixture of calcium to just suddenly turn into a hard rock?
By acting as a microscopic scaffold. The OC17 protein essentially clamps onto the calcium carbonate particles, forcing them into a highly ordered, tightly packed lattice. It basically triggers the crystallization process.
Oh wow, so it physically arranges it.
Yeah, and once the nucleus of the crystal forms, the OC17 detaches and moves to the next spot to repeat the process. It does this with astonishing efficiency, like building a fully formed, rigid eggshell in just 24 hours.
And here is the crucial patch from the research, the thing that just turns the whole paradox on its head. This OC17 is only found in the ovaries of chickens and their close relatives. Nowhere else in nature.
Nowhere. The causality of the structure is undeniable here. The amorphous calcium carbonate desperately needs OC17 to crystallize into that specific hard shell. But OC17 is a proprietary protein manufactured only by the chicken's physiology.
I was really trying to wrap my head around this earlier, and it's basically like a highly specialized 3D printer.
Okay, yeah.
Imagine you have all the plastic element in the world. That's your raw calcium carbonate. But you physically cannot print this one specific, intricate object without a unique, patented nozzle.
I like that.
And that nozzle is the OC17 protein, and it is permanently attached to the inside of the chicken. So for a chicken egg to even exist, the chicken's biological machinery must have come first.
That is a perfect analogy. You simply cannot separate the final product from its specific biological manufacturing process.
It's crazy.
It is. And this apparent contradiction, where evolution is loudly declaring the egg came first, while structural biology insists the chicken came first, it's beautifully reconciled by researcher Abdelkrim Richetti. He points out that the paradox is solved simply by context.
The general rule versus the specific mechanism.
Exactly. Evolution dictates the general rule.
Yeah.
The reproductive cell, the egg, predates the organism by 1 billion years.
Right.
But structural biology provides the specific mechanism. The physiological anatomy of the chicken is an absolute prerequisite for the formation of the chicken egg.
So they're both right.
Yes, both answers are completely scientifically valid. They just operate at totally different levels of biological analysis.
Okay, that is an incredibly resilient protein. But sitting here, I can't help but wonder, like, outside of making a really durable breakfast, how does a heat-proof eggshell protein actually impact human beings today?
Wow.
Because going through these sources, finding this OC17 protein unlocked what I can really only describe as a biological superpower hidden inside the eggshell.
Oh, definitely. This is where the deep dive moves from, theoretical philosophy to highly applied real-world science.
Yeah.
A paper published in the journal Soft Matter looked really closely at what happens to OC17 after it catalyzes the shell.
Okay, what happens to it? Does it just wash away?
That's what you'd think.
Yeah.
But during that rapid crystallization process, OC17 doesn't just do its job and leave. It actually becomes incorporated and trapped directly inside the calcium carbonate crystal lattice of the eggshell.
So it gets walled in by the very structure it just built.
Yes. And when it is trapped in that crystal lattice, the protein becomes virtually invincible to environmental hazards, particularly extreme heat.
Really?
Yeah, the researchers extracted this crystal-bound OC17 and heat-treated it at 50 degrees Celsius, that's about 122 degrees Fahrenheit, for 60 hours.
Which, for a typical protein, is a death sentence. I mean, if you crack an egg into a hot pan, the proteins denature immediately, they turn white, they unfold, they lose all their biological function.
Exactly. A normal protein floating in a solution would completely denature and unfold under those harsh conditions.
So how do they know it survived?
Well, the researchers used a technique called thermogravimetric analysis, or TGA.
Okay, and how does TGA actually prove the protein survived?
TGA essentially measures the precise weight of a material as you subject it to extreme increasing heat. When a protein is destroyed by heat, it breaks down, burns, and turns into gas.
Meaning the sample loses physical mass.
Right. The weight drops. But the TGA analysis showed something incredible. The organic material, the OC17 protein trapped inside the eggshell's crystal lattice, maintained its weight. It was protected up to 250 degrees Celsius.
250 degrees Celsius, that is nearly 500 degrees Fahrenheit.
Nice.
The protein just sits in there perfectly fine while the world basically burns around it. So what does this actually mean for us?
It addresses one of the most critical logistical challenges in global healthcare right now, the cold chain crisis.
Oh, right.
Many of our most life-saving vaccines and protein-based therapeutics are incredibly temperature sensitive.
Like the mRNA vaccines.
Exactly. Think about recent mRNA vaccines that required ultra-cold storage, specialized freezers, dry ice just to transport them. They require constant, unbroken refrigeration from the moment they're manufactured in a lab to the exact moment they're injected into a patient's arm.
Which is just a huge, expensive problem, especially in resource-limited areas. Or, during a global pandemic where you're trying to ship billions of doses around the entire world.
It's a logistical nightmare.
If a truck breaks down or the power goes out at a rural clinic, the medicine is just ruined.
Yeah, the cold chain fails frequently and with devastating consequences.
Yeah.
But scientists are looking at biomimicry to fix this.
Copying the chicken.
Yes.
Oh.
If they can mimic the chicken, taking these delicate, temperature-sensitive vaccines and incorporating them into cheap, room-temperature calcium carbonate crystals, just like OC17 is trapped in the eggshell, we could theoretically ship life-saving medicines anywhere in the world, completely outside the cold chain.
So a box of vaccines could just sit on a hot tarmac in the sun for days and remain perfectly viable in like a powder form.
Theoretically, yes.
I love the vision. It sounds like science fiction in the best way possible.
Okay, it does.
But I have to play the skeptic here for a second.
Please do.
I admit some confusion about the logistics of this, because it's one thing to trap a delicate vaccine in a microscopic rock so it survives shipping. But how do you get the vaccine safely out of the crystal once it actually reaches the clinic?
That's $1 million question.
Right, because usually you dissolve calcium carbonate with an acid, but wouldn't pouring harsh acid on the delicate vaccine just destroy the 3D structure of the medicine anyway?
That is a highly astute question, and it's the exact hurdle the researchers had to clear. Acid dissolution would indeed risk denaturing the therapeutic protein, completely defeating the purpose of preserving it in the first place.
So what did they do?
The solution they found is a chemical agent called EDTA.
Okay. How does EDTA work without using acid?
Well, EDTA is a metal chelating agent. So instead of aggressively burning the crystal away like an acid does, it acts more like a microscopic chemical claw.
Oh, a claw.
Yeah, it binds specifically to the calcium ions, gently and slowly, pulling the calcium carbonate crystal apart at a totally neutral pH. It completely avoids the need for any harsh denaturing acids.
Oh, wow. So it essentially unbuilds the house brick by brick to get to the blueprint safely inside.
That's a great way to visualize the mechanism. Yeah, they tested this process using a model protein called BSA. They locked the BSA in the crystal, exposed it to high heat, and then dissolved the crystal using the EDTA chelating agent.
And did the protein actually survive the unboxing?
It survived flawlessly.
Really?
Yeah, they verified this using two highly advanced techniques, circular dichroism and tryptophan fluorescence.
Okay, yeah, you're going to have to translate those for me.
Sure. Think of a complex protein like a pop-up tent.
A pop-up tent, okay.
Circular dichroism checks the secondary structure. It essentially checks to make sure the springs and hinges of the tent are still coiled correctly and haven't snapped.
Okay, so the hardware is intact.
Right. Then, tryptophan fluorescence checks the tertiary structure. That makes sure the entire tent still pops up into the correct three-dimensional shape.
So the hinges still work, and the whole structure actually still stands up when you deploy it.
Exactly. Both of these tests prove that the 3D structure of the protein emerged from the crystal completely intact and ready to function. It is a slow-release, perfectly safe unboxing of the molecule.
That is so cool. It's amazing that this blueprint inside a chicken can, potentially save human lives today.
It's revolutionary.
But what's even wilder to me from the sources is that we actually carry a broken version of that exact same egg-laying blueprint inside our own human DNA right now.
Oh, yeah. This is a fascinating evolutionary relic. We really have to remember that all mammals evolved from those ancient egg-laying amniotes we mentioned earlier.
Right.
And because of that deep shared ancestry, human DNA still actually retains the specific genetic instructions for creating a yolk sac.
Which is just wild to think about. I definitely do not remember having a yolk.
Well, you did, very early in your embryonic development.
Yeah.
But it's largely vestigial in terms of actually providing you with any nutrition.
Because we have placentas now.
Exactly. Because mammalian embryos evolved to receive their nutrition directly from the mother, through the placenta, the biological need for a yolk just faded away. Over millions of years, the specific genes responsible for manufacturing the yolk itself accumulated mutations and became what we call pseudogenes.
Pseudogenes. So they are basically like broken lines of code in our biological software, just sitting there taking up space on our hard drive.
You could certainly look at it that way, yeah. Evolution is highly efficient, but it doesn't always erase its old drafts.
Right.
These genes are totally non-functional in us today, but they remain physically etched into our genome. They are an undeniable physical receipt of our egg-laying ancestors. We literally carry the history of the egg within the nucleus of our own cells.
A biological receipt. I love that. And we aren't just passively carrying the legacy of the egg either. We're actively continuing to evolve its purpose.
How do you mean?
Well, according to the research, scientists in Japan are currently crossing genetically modified chickens to lay eggs that act as little living biofactories.
Oh yes, I saw that.
Yeah, they are engineering the biology of the chicken so that the eggs they lay naturally contain specific cancer-fighting and immune-boosting proteins. We are literally reprogramming the ancient technology of the egg to heal ourselves.
It is a profound testament to the versatility of this biological structure, really. It has gone from a billion-year-old, single-celled marine organism experimenting with multicellularity in the ancient oceans to a modern vessel for next-generation cancer-fighting therapeutics.
Incredible. So let's bring this deep dive full circle for you. The next time you are at a dinner party or, you're in a meeting trying to break the ice, or a kid raises their hand and asks you that ultimate classic riddle, which came first, the chicken or the egg? You now have the definitive, scientifically backed answer to give them.
Broadly speaking, from an evolutionary perspective, the egg predates the chicken by hundreds of millions, if not a full billion years. So the egg definitively came first.
But specifically speaking, from a structural biology perspective, the unique 3D printing machinery of the chicken, that incredible OC17 protein, absolutely had to exist first to build the chicken egg. So the chicken came first. You get to be right no matter which way you argue it, as long as you know your context.
And knowledge is indeed most valuable when it's understood and applied in its proper context.
Well said. But before we go, I want to leave you with one final kind of mind-bending thought to mull over. It comes from the zoologist Jules Howard, whose work was in our stack of sources today.
Oh, this is a great concept.
Right. As humans, we have this intense bias. We naturally assume that animals use eggs to reproduce themselves.
Yeah.
We see the animal as the main character of the story.
A very anthropocentric view of biology.
Yeah.
Assuming the adult, moving, breathing creature is the pinnacle of the entire process.
Right. But if you look at the vast, sprawling timeline of planet Earth, a timeline where eggs existed hundreds of millions of years before complex animals even had legs or eyes or brains, what if we have the relationship completely backward? What if an animal is really just a temporary fleshy vessel that an egg uses to make more eggs?
Now that
Yeah.
is an entirely different kind of infinite loop.
Just like an MC Escher staircase you can never quite get off of. Keep questioning everything, and we'll catch you on the next deep dive.